categorical data versus quantitative data under the microscope. First of all, categorical data is defined as names or labels that are not representing measurements or counts. While in the lab, you observe different specimens. These specimens can be labeled under various categories. Example categories include prokaryotic cells, including archaea and bacteria, or eukaryotic cells, often including blood or tissue samples. Now, there are two types of quantitative data. Discrete data, which is a set or finite number, while continuous data is changing or there are too many possibilities to count. Discrete data. It is defined, once again, as quantitative data that is a set or finite number. When specimens are viewed under the microscope, they are most commonly stained. This process usually kills the sample, stopping any more growth, which would produce a finite number of cells within the slide. Below you'll see a gram stain. The gram stain goes through a heat fix, which plasters the cells to the slide, but also kills them. Therefore, there would be a definitive number of cells to be counted. Continuous data. Defined as data where values are not countable. The data is always changing or can be altered. Often in the lab you have to grow specimens to see change or provide further samples. When growing these samples, the growth is not controlled. It is a varying number of cells in the plate until someone in the lab manipulates it. As you'll see below, the culture produced small colonies within the first stage of the growth cycle and would continue to grow until someone in the lab manipulated it, therefore continuously changing. And that is categorical data versus quantitative data under the microscope.